The section of notes we're going to be covering today are parts of the microscope. So please find that page from your notes. Make sure you have something to write with. We're going to start off by looking at the diagram. So make sure you're on page 2 of the notes. It has the diagram of the microscope there where some of them are filled in, some of them are blanks. You'll notice that each line has going to it a description of what that item is and then the name of that item. Please make sure that you are studying these terms so that way when you hear directions that say to raise the stage all the way up, that you know what we're talking about. Okay, instead of calling it the platform, make sure that you're calling it the correct name. Alright, we're going to start at the top left-hand corner. You see that the description has been given that says contains the lens to increase magnification, usually 10x. 10x means 10 times magnification. So it's a 10 times larger image than what you normally see. And this is called an eyepiece. Okay, the eyepiece is right there at the top. So you see where it is on your diagram. Remember that you can pause the video at any time you need to in order to fill in these notes. Next, below the eyepiece description is the arm. This is what supports the objectives and connects them to the stage. This is the arm. Below that, the description we've been given is that it moves the stage up and down approximately to correct distance. This would be the course adjustment knob. It's really important that you see that this says move the stage up and down approximately. This is a larger, quicker movement, approximating where the distance needs to be. In this diagram, it is a separate knob. I will say on our microscopes, these two knobs are together, but it is the larger knob. Next, it permits finer focusing by moving the stage in smaller increments. So the first one was approximate, this one's finer. This would be the fine adjustment knob. Then the last one there on the left side is the base. This supports the microscope. Next, starting there at the top right hand side, is the body tube, and this is what keeps the lenses at proper distances from one another. So this is the tube that's connecting the eyepiece to the objectives there on the microscope. Below that, we have the description that revolves to allow changing various objectives. This would be the revolving nose piece. So it holds each of the objectives and it is revolving. Revolving means to turn. Below that we have objectives. These are what magnifies the image. Some microscopes have 2, some have 3, some can have 4, and they all have different measurements on them. Like they'll say 10x or 40x or 100x. Those are all the magnification power. A 10 times magnification is only going to magnify that object 10 times larger than its normal size, where an objective of 40 is going to magnify it 40 times its normal size. Next description we have is that it holds the slides in place, and these are the stage clips. Below that is the stage. This is a surface where slides are placed. The next description regulates the amount of light going through the stage. This is the diaphragm. The thinner the object, the less light you can allow through the diaphragm. The thicker the object, the more light you're going to need to allow through the diaphragm. And last is the light source. This is what passes the light through the stage. The light source can be a bulb that is exposed, or it can be underneath something, but it is lighting upward So that way there's light going through the diaphragm and to the specimen. Okay, so you should be on the next page for the microscope notes. And the first question is, how do we study cells? We have to use microscopes. We can't look at them with just our plain eyes because they're too small. We have to use the microscope to magnify them. There are two important characteristics of a microscope. One, the magnification. This is the ability to make an object appear larger. Second is the resolution. This is the capacity to distinguish between different objects that are close together. Okay, so a higher magnification makes it appear bigger. The resolution is going to be that sharpness that's going to let us know where one part of the object ends and the other one begins. There are a couple of different microscopes that we're going to talk about. The first one is a compound microscope. This uses two or more lenses to magnify an object. To use a compound microscope, the specimens must be very thin. Okay, these are the ones that we use in class. The very thin part you'll know because the slides that we'll use, you can see they're like paper thin specimens with a cover slip on top of them. These microscopes have limited magnification, but they're cheap. This is why it's nice. Okay, now when I say cheap, I'm not talking about like something that you can just pick up at Walmart. Like these are expensive microscopes, but compared to the other kind of microscope we're going to talk about, these are relatively cheap. Okay, so here's some images of a compound microscope here. Once again, these are ones that you've seen before, probably used in science class. They've got the eyepieces connected to the microscope there and the objectives that rotate. You can see images through slides, and just normal specimens are what can be looked at here. The other kind of microscope that we're going to talk about is an electron microscope. In these, you're going to pass electrons over or through an object to magnify it. These are really nice because they can have a really good resolution at a high magnification. So that means you can make things that are really tiny. You can make them really large and still keep a lot of sharpness within that image. The only downfall, they are very expensive. Okay, like another class once before, we looked up what a used one would cost on the internet, and like a really old used one was still like tens of thousands of dollars. Okay, for our electron microscopes, you need to know that they can only be used to observe dead cells or non-living structures. Okay, we're passing electrons over or through them, so if you take an object that's not living, this can cause multiple problems. So the structure has to be dead or non-living. It has to stay completely still. Next, there are two types of electron microscopes. One is the transmission electron microscopes, or TEM microscopes. These have electrons that are passed through a specimen. Next are the SEM, the scanning electron microscopes. In these, the electrons are passed over the surface of the specimen. So TEM goes through and SEM goes over. So think about T for through and S for surface, so going over the surface. So there on the right hand side you can see this is an electron microscope. It's huge. Yeah, it's not like just a little piece of that. That whole entire contraption is an electron microscope. Now you can probably see why these things are so expensive. But look what's so cool about them. These are a couple of images that were taken from electron microscopes. You can see the top left one, that's a dust mite, and how sharp that image is. Isn't that just amazing what it's able to do? The bottom left hand image looks like it's some sort of retrovirus, probably. It's a teeny tiny cell that they've been able to blow up and create that image. So once again, these electron microscopes are so cool, but there's no way that we're ever going to have one here on our campus. but we can at least use images that were created from electron microscopes from the internet. Okay, next we're going to look at microscope use. These are rules that you need to apply when using microscopes. Please make sure you study over these rules. I constantly see students using microscopes incorrectly, and guys, it can mess up your experiment, it can cause damage to the microscopes, and it can just be a hassle for those that use the microscope after you. So please make sure that you're looking at these rules, that you understand them, and that you follow them. Okay, first, you always start on low magnification. So if there's an objective with a 10x or a 4x, you're going to start with that one first. You can use the coarse adjustment knob now. Remember, that's the one that moves the stage up and down very quickly. Okay, the reason that you do this is because the field of view is larger on a low magnification. Your field of view is what you see when you're looking through the lens. So when you're using the low magnification, you have a larger view of the object. This allows you to get your specimen centered a lot easier. Nothing is more frustrating than trying to find that specimen, like if your slide isn't exactly centered, it's if you're on a really high magnification, you're moving that slide just the tiniest bit, and it's making like a huge jump in your field of view. So use it on the low magnification, use the coarse adjustment, move your slide around until you can find the specimen within your field of view. Okay, so you're always going to use the coarse adjustment first with the lower power objective. Once the specimen is in view, then you're going to switch to a higher power objective. You're never going to use the coarse adjustment with a high power objective. So once you bump it up from the 4x or 10x to maybe like say the 40x or 100x, then you're going to switch to using the fine adjustment knob. Okay, you don't want to move that stage a lot. You just want to move it just a little bit to be able to perfect that sharpness. When you're finished using the microscope, you're going to lower the stage using the coarse adjustment knob. Move it down real quickly. Then you're going to leave it on the lowest power objective and turn off the power. So I should never see microscopes left with the stages all the way up or on a high power, or even more frustrating, the light on. Make sure that you've got the stage all the way down, that you've got the lowest power objective switched over, and that you turn off the microscope. Okay, last thing we're going to do is look at the magnification of the microscopes. There are some calculations that are involved when you look at the magnification that you're using in an experiment. You need to know that you multiply the eyepiece and lens magnification with the objective lens magnification. So for example, the eyepiece magnification is 10x. So remember, the eyepiece is the little thing that you look through, and it's most of the time it's a 10 times a 10x magnification. So in this example, we've got our eyepiece magnification of 10x, and the objective magnification, remember the objective is the thing that you're switching there on the microscope, is 4x. So what is the total magnification? You're going to multiply these two numbers and say that your total magnification is 40x. So using those two items together, you are magnifying your specimen 40 times larger than its normal size. Okay, next, the eyepiece magnification is 10x and the objective magnification is also 10x. What is the total magnification? Okay, you should have said 100x. This is going to conclude our notes on microscopes. Now you're going to go do some practice with what you learned here from these notes. So good luck, and I will see you back here to continue on with the next set of notes.